We do want to do research that stands out, not as an individual, but our research somehow has to survive the time for at least for a while, and be seen and be noticed and be an inspiration for other work. If nobody cares about your research, it would also be quite depressing, I think.
Welcome to Called to Science, where we delve into the untold stories of the scientists who are pushing the boundaries of the life sciences. I'm your host, science journalist Brad Van Peerdon, and today we talk about the joy of discovery, the importance of finding the right question, and tips on how to catch a horse. Welcome, Stein Aerts. Not many of us have wondered what all the genes in all the cells of a fruit fly are doing at any given time. But you did, and you created the cell atlas for the widely used model organism Drosophila. Your work also uses techniques from computer science, data analysis, and mathematical modeling to describe biological systems and relationships. And you're leading the way at VIB for the use of AI in biological research. However, before you went digital, as I understand it, you had put on your explorer cap and spent some time in the jungles of Ecuador. Welcome, Stein Aerts, to Called to Science.
Thank you so much.
You did go to Ecuador. That was just traveling.
Traveling.
Yeah. So any stories you can tell us from there? Did you do a lot of like trekking in the jungle?
Yes, also. That's a long time ago since I thought about this, but it was a very, I've traveled a lot in general, alone, when I was a student for usually a long time. I used to even arrange my travels and book my flights, not knowing if I would have passed. And then there was not really WhatsApp at that time, right? Mm-hmm. So then I would have to find a public phone wherever in some village.
A phone booth, something that doesn't exist anymore.
Exactly, a phone booth, exactly. Put in some coins and call my mother to ask if I passed my exams. And that was the case, luckily, so I could stay. And so I was doing a couple of times voluntary work in Ecuador, for example, spending a couple of weeks on some commune place where we would help. What were we doing actually? Maintaining pieces of the cloud forest with. Just some people had to be there and to maintain a bit of the plant organization, helping farmers with certain types of plants and so on. And then you could just stay there for weeks with other international students and not have to pay anything. You didn't also earn anything, but you could just stay there. You had a place to sleep. You could make friends and visit the jungle and the cloud forest. And then I also passed from Ecuador to Colombia, just backpacking. And then later with my wife, we also continued traveling, also with the kids. So now we went together to Venezuela and Mexico. And yeah.
Interesting. I did something similar, the backpacking trip when I was in my 20s, but I did Brazil. Traveled all around Brazil.
Never been to Brazil. That's high on the list.
I really liked it. And I'd spent some time in the Amazon, in the jungle as well. And I'm curious, I was already interested in biology when I did that trip. You were there doing biology as well and doing some volunteering stuff. What did you think of the jungle when you first saw it? For me, the diversity, the examples of evolution that you get there, these things that are perfectly adapted, and the interconnectedness of all these life forms. For me, it was really quite mind-blowing as someone that was interested in biology.
Yeah, absolutely agreed. The plants so when I was telling that I chose this genetics option, there was also a second option, a minor you could choose, and I chose plant genetics and plant diseases. And so that was very interesting to indeed have that as courses and then go into the jungle. And so plants were always the first thing I saw, rather than, animals are always harder to see, of course, to find. But birds, toucan was always one of my favorite to try to spot. A fantastic experience. In Ecuador particularly, it was great because it's a bit of it's called cloud forest, so it has yet another type of atmosphere, and it's very, very enjoyable. Not so hot and humid, but still fantastic.
Any close calls with some of the bigger animals, or any
Not
exciting
really.
stories? No, it was
No.
just plants.
Plants,
Which
I
could
think
also be dangerous.
Yeah, the most exciting story I think is that we would always do horse riding without saddle or anything, and there were horses around, like they owned some horses, and you had to like kind of find them where they were, because they were more or less located in some area, but not really. And then you had to find them and then try to get them to you with some salt or something. Yeah, and then we would throw a rope over the back, and then the horse, do you know that the horse thinks it's around its neck, so then it stays still. Yeah, that's a trick to catch a horse. You just throw a rope around the neck, and then you can bind it, and it would just be a rope. And that's why I learned there to ride a horse. Just jump on it and do something, and then you can just. We would ride it for hours in the jungle. To go up to the cloud forest was very high up, and we just take a horse and then go up. Yeah, that was very fun.
That's incredible.
That's a long time since I thought about this.
Wow.
That's you bring the nice memory.
Okay, well, good. Well, I learned something too now. If I ever need to capture a horse,
You can
I
call
guess.
me, or you know the trick. It's not so difficult.
I won't need to find a phone booth to call you now, so
Yeah.
that's good. Well, let's back it up then first. And you grew up in Belgium.
I did.
What was your introduction to science, or was there someone in your life that introduced you to science, or school that really kind of stoked your passion for this and maybe put you on the path towards being a researcher?
Well, if I think back on high school first, maybe we did have a small group of people who were following mathematics, with some more mathematics, like 8 hours a week or something, with a small group. And there was a fantastic teacher. Physics as well was great in the last 2 years of high school. And it was clear for me already that I was going to do some scientific studies. And then I chose for bioengineering. With after a few years, you can choose different options, and then it was called cell and gene biotechnology, so genetics basically, but from an engineering, bioengineering perspective. And so gradually, this became more and more a passion to do genetics and to understand genetics. And indeed, then during the time I was studying bioengineering, I was really mostly interested in the molecular biology, biochemistry, genetics courses, and I was very, very happy that I chose that option. And I still think this is one of the best programs to follow. It's you start with technical material in the bioengineering, but then the genetics option is very, very specialized. So you really, really learn a lot about molecular techniques, and this I still carry with me, and that's also in my lab at the moment. We have a wet lab and we do computational biology, but in the wet lab part I have as much fun as in the dry lab aspects. So we still do quite some advanced molecular techniques and technology development, and this still stems from my earlier education there. The technical stuff of the engineering with the molecular biology, this was the foundation, I think. And then later, the computational part was added to that.
Well, let's turn specifically to your career again in research and in science. And I wanted to ask you about any moments where you first kind of had that feeling of discovery. For me, when I did an undergraduate project on amphibian rhinoviruses, I remember that something that then became routine, but the first time that it happened, when I got that PCR band to show up and it confirmed that, yes, the virus I was looking for was present in our population, etc., was just a mind-blowing moment for me, and like really opened the door to, wow, this is discovery. This is, what's possible with science. Did you have a moment like that, or was it more gradual for you?
When I finished my studies, I first wanted to do a PhD, but then in the end I didn't, and I went for programming because I liked programming during my studies, but we didn't really have a lot. And this was 1999 or something. So it was called the internet bubble. And yeah, companies were hiring anybody to program, even people who didn't study computer science. And so they hired me as a bioengineer while I could actually go and study computer science in Brussels. And so they would allow me several days or 1 day a week at minimum free to study. And so then I started to study computer science. That took me 2 years. And then I decided to do a PhD, because when working in a company for software development and so on, I was always looking on the internet for, microarray data analysis papers. I was reading papers in my time, and I realized, what am I doing here? And this was really the time, the genome was just sequenced, microarrays were invented, and you could just profile expression of all genes. And I really thought a lot about this, but I can not remember why I was attracted to this particular technology. I found it fascinating. Of course, I studied genetics. I had every project I had to choose was always about gene expression. So I really wanted to know how genes are regulated from the beginning. And then this technology came up, and I was reading, and then I wanted to do a PhD on that. And then I finally found that at ESAT, Electrical Engineering department. And so to come to your question, like this then allowed me to really go deep into computational analysis of genome sequences and of microarray gene expression data. And at those times, it was one discovery after another almost. We published a lot of papers in a short time, because you could, this was all new and unknown. And all these discoveries, I still remember very, very vividly, all these scientific discoveries. And then when I started my postdoc, I had another moment like that I always remember very well. The first time I made a transgenic fly with an enhancer reporter that I predicted from sequence data and from microarray data, again, to be active specifically in a particular cell type at a certain moment during the development of the eye. And this is a very important gene that starts making neurons, called the proneural gene. And I was working for my postdoc 5 years on trying to figure out which genes that this proneural factor controls. And therefore, I spent a lot of time making my prediction as good as possible, because I knew I was going to spend 6 months testing 20 or 30 of those. But then the first I tried, it worked fantastically, yes. And so the GFP signal under the microscope of the eye disc, right at the place, I fell from my chair. This was spectacular. So that's why also microscopy, we still have been doing such experiments all the years in my lab as well, testing activity of DNA sequences, where they can activate genes and so on. And that usually goes with microscopy, and that's still very, very pleasant. Yesterday we had in our Slack channel a video of the first time we tried such an assay in zebrafish with a green heart, and the heart was beating and it was all green. So the experiment worked, and we could design heart enhancers for zebrafish. And every time it's almost daily that we have these moments of, we're always eager to find them, of course. Maybe not daily, but I would say almost weekly to have.
Weekly is a pretty good success rate. Is a pretty good success rate.
Yeah, if you work with a lot of data, there's always some new, plot or visualization or enrichment, that a signal, right, that we find. Mm-hmm. Yeah.
I think it's having those visual cues, like the glowing green from the GFP or like the PCR band. There's something about that, when you can you see it happens, and it's like it just screams success. That's so cool.
It screams success. Yeah. Exactly. Yes, we were right. It works.
Yeah. So it's time to dig a little bit deeper in this Dare to Ask, and here I'm going to give you some questions that other people have come up with. These are questions from the VIB community. What's the secret to standing out as a scientist at an institute like VIB?
Oof, I don't like to think about standing out. I've never had any plans, right? In my PhD, not in my postdoc, not. I didn't just continued with the things I liked to do. So standing out is I don't really like this, as a goal too much. Of course, there is a aspect of if you can do a number of impactful discoveries, then there might be opportunities to have more, let's say, to find people that want to come to your lab because they've seen things that you have done before. Right? For a young PI that starts a lab, it's not so easy to find people, and so particularly postdocs. So if somebody does a PhD on an interesting topic and then finds your lab because you have done this or that discovery, then may apply or may contact you to come and join your lab. I think this is really very, very nice, right? So we have done a couple of very good discoveries, I think, and this way it's a kind of standing out in the sense that being noticed for your research, i.e. it has impact, it is seen, it is appreciated, and we like the way that you have done the research, we like the way that you describe it in your papers. So can we have a chat about joining your lab? I mean, that's fantastic, I think. So that is, it does matter, right? I'm not going to say it doesn't matter. We do want to do research that stands out, not as an individual, but our research somehow has to survive the time for at least for a while and be seen and be noticed and be an inspiration for other work. If nobody cares about your research, it would also be quite depressing, I think. So in that case, you kind of want to stand out with your research to the degree that it is appreciated and noticed, because there's a lot out there, a lot of stuff, a lot of papers, a lot of researchers. So it's daunting if you think about it, but we don't think about it. It comes as it comes. It's in retrospect nice that we have done some impactful things so that we stand out, but we didn't do it for that, you see?
I understand. And so now I'm actually going to pick one that maybe pushes you a little bit as well in this same vein.
Right.
If you could pick one thing to define your legacy at VIB, what would it be?
Pure scientifically, it would be nice if our legacy would be a significant contribution into understanding how the genome works and how it controls our cells in our body, how that is encoded in the genome sequence and what we can do with that. But of course, there's also some meta aspect of it, and legacy is a lot to do with the people in the lab that go out into the world, and I have a lot of PhD students who are doing fantastic postdocs around the world, and that's a very, very, and when you see them again at conferences and discuss how it goes, and when we had our 10-year lab party, I invited all the alumni, and many of them came, except somebody in Australia, couldn't make it, but it just flew in, It was so nice to see, and the legacy of the people and the research that they carry onwards, and what we just talked about, the process of doing science, that there are alternative ways, and that through computational approaches in general, we can address very important, hard biological questions. That's hopefully a little piece of that. I think we would be nice to have contributed to the community.
If after listening to this Dare to Ask, you find yourself with a question you've never dared to ask, let us know in the comments. And who knows, maybe your question will end up in the next episode. So we talked about all of these moments where you get the success, it works, you see the color, you see the PCR band. What about all the times it doesn't work? How do you maintain your drive to keep going?
How I maintain it, I don't know. I do maintain it. I've seen that. This is really something that's there. The drive to decipher the genomic regulatory code is very strong. I have it very strongly there. It's also really something, as I told you, super fascinating and important. I also think that we understand that. And at every question how nice it can be, indeed, we have failures. Technology and wet lab has a lot of failures, and their persistence is really from another level, I think. And computational biologists in the lab may not always realize this so well. They also have failures, and they also have to persist. But you can go to sleep and the next morning try a new approach, a new piece of software or a method that somebody published on your data. Right? Your data is not, you don't have to throw it away. If the data is good and you don't find the answer yet, you can continue searching. In the wet lab, if something fails, you have to start over. And if it fails many times, starting over many times means weeks and weeks and weeks go by. And then the persistence is sometimes harder. And you also, especially for developing new protocols and new technologies, we are sometimes a bit crazy, I think, that we do wet lab, molecular biology innovation, and technology innovation. We published a few new techniques that the community can use now, rather than using something that exists. We're just interested, this is by my bioengineering background, right, to develop something new and new computational techniques. And then if it's about new technique, the idea is about, if we do it this way, it may give us better results or better signal to noise, or it can be cheaper and so on. But it's hard to predict, right, if this would really work out. So then the development of technologies, there's been a lot of failures. I try to let's say, convey my own enthusiasm to them, and it often works. And I also explain them, right, really the reality of the matter, that this goes by ups and downs, and sometimes it's hard, and we have to persist, or we take an alternative route, try something else, and keep going. It's really part of how we do it. I don't really know, but we're doing this. It is inherent. If you don't like that type of problem solving, this is something that people in my lab, and I think many scientists just are attracted to solve a problem, and so it doesn't have to be simple. If your puzzle is hard, then you may even like it more. And of course, you don't solve it in an afternoon, so it doesn't work, but you still persist. That's a very good definition of how our days go about, I think. Mm-hmm. But the goal is quite clear for us often, where we want to get. So then we have to find a workaround as engineers, find a workaround, do it in a different way, turn things upside down. Yeah.
Right. I think it's for a lot of people outside of science, and maybe new students coming into science, they kind of maybe think of it as just, plucking these facts out of, you just do an experiment, you get the fact, and then now we know what it is, right? But it actually is a lot more problem solving, trial and error. You actually have more failures probably than successes, or the ratio is.
Yes, absolutely. You have more failures, and a lot of the time is spent on finding the question, especially for us. So in computational biology and data-driven research, we usually don't start with an hypothesis. Right? So that's also a particular way of doing science. It's another procedure in a way, another type of process, where we have a feeling that by generating such and such data, we may find something interesting in general, or around that problem. Something interesting may come up, but we don't know yet exactly what it is. And then by going through the data and modeling the data, we are coming closer and closer, hopefully, sometimes further away, to some very interesting question. So we are finding also the question and then the answer. Mm-hmm. And that's a kind of a search, a space search algorithm, or that we have to go through ourselves. This is different from hypothesis-driven research, where you think, I think this protein binds to this protein. That's what based on literature and my experience and so on, I hypothesize that, and now I'm going to test that. Do you know this Night Science podcast?
No, I'm not familiar with it.
Yeah, so they talk about this. There's a podcast that is about this distinction between what's called day science and night science. It's very interesting. It comes from Jacob and Monod, where in day science you can say, okay, this is how I think it works. So I have to do an experiment, and then my readout is the fact that you allude to. It is indeed, it binds or it doesn't bind. Night science is when you have to come up with this ID. And so this doesn't mean you have to do it in the night. So computational research is the majority is night science, because we don't really know yet where we're going. And through computational modeling and data and other data and ideas and talking, we are carving that out.
Yeah, that's interesting, because it is like you're searching for the question.
You're searching for the question.
Excellent. So I want to talk about computational biology. This sort of, to me, as a naive person, it really feels like the two aspects, sort of this programming, this digital, the software kind of element, and then the living systems, the biological systems, seem totally different. Do you see them as one is the computational side is just a tool that helps you understand the other one, or are they more similar than one would think?
Another interesting aspect about the question that we have in the lab and that I've had since before my PhD, I think it's the most fascinating question in life sciences. Maybe in physics you could argue that black holes are interesting to study. I'm with you, but in life sciences, the question of how DNA sequences encode life, encode all kinds of different life forms, the same DNA, right? It's A, C, G, and T, and you put them in a different order. So a sequence, literally, that's the sequence. It's the sequence, the order of the nucleotides. And you can make, is this a real plant? You can make this plant, if that's plastic, you couldn't. Or a pea, right? Or a squirrel. And you change the order, you make a fruit fly. You change the order, you make a human. And that's really fascinating. And then, obviously, whenever any life form has multiple different cell types, gene regulation evolved, because the same genome sequence has to be differentially regulated between these different cell types. They extract differential information from the genome sequence. And that is a very, very computational problem.
Yeah, it's interesting, because you start to then see, cells maybe as little biological computers. They're doing these computations, and there's all these sort of algorithmic problems that go into it. But there's an element of creativity, obviously, that has to come in too, where like you said, you need to ask the right questions. So you have to understand how to use these powerful tools in a way to answer these fundamental questions about life, right?
Sure. That's, I dare to say that's our speciality, right? For this field, in this area, with a lot of experience and a lot of passion and drive to ask the right questions and to use the right techniques and to find hopefully the right people, because that's where it depends a lot on, right? Having people who share the passion with you, who are also interested in this so much and who can carry this forward and who can spend their time on solving new questions and being able to change rapidly. It's also one of the things I noticed. So I had started my lab in the University of Leuven. It was in 2009, I think. And in 2016, 2017, I got the opportunity to become group leader at the VIB, where the funding system is a little bit different, where groups can get funding in advance, you may say. I think this is the best system of science funding ever. So you put trust in somebody. So I trust you. Why? Well, because you have already a proven track record. You have done very important discoveries. So here's funding, and you can do whatever you want. But in 5 years, so every 5 years, there's an evaluation, a check, right? In 5 years, you can come and explain to experts in the field, to peers, let's say, what have you done? And if it's good, you can continue. This means that at any given moment, with this funding, you can make a new decision. You can buy an instrument or start a new technique or recruit somebody new and switch overnight and start something new overnight. While classically, you would have to think about a grant proposal, right? Then write the grant, submit the grant, wait for the grant, gets rejected, submit it again. So 2.5 years later, that's not really...
And the moment may have passed.
The moment has passed, long time.
Yeah.
So being able to, I think this is a nice way of, it's in general true in science and in universities, the principle of, the trust is there. We trust you, do whatever you want. And we'll trust it's going to be important and meaningful for society in the end, at the end of the day. This is the beauty of the job. We are completely free. We have no boss. And then in addition, that's a university principle already. And in addition, in the VIB, we have the opportunity to do excellent research and to adapt very quickly to new situations. And when you combine that with a new question, it can be fireworks. Yeah.
So as you're looking to recruit these young scientists to come join VIB, be a group leader, what would you say to them to convince them to join you?
Yeah, we have talked a lot about the research in my lab, but indeed we are have started up a new center, AI and Computational Biology Center in the VIB, where at the moment we are recruiting new group leaders, and I'm trying to find group leaders that have a similar idea of using AI, but on biological problems. So it's probably going to be a mix. We have a beautiful opportunity to recruit group leaders to share a new center with. And for me, in an ideal case, some of them would be on the spectrum a little bit more computational, computer science, working on the algorithmic and AI methods, but with some application to biology and some notion of biology, but then more and more, and the majority, let's say, to have an actual biological question like we do on the genome sequence. It can also be on the genome sequence. I wouldn't mind to have some more people to try to decipher this code using AI, but there's, of course, multiple additional layers in biology. And if the group leaders who would like to, or looking for positions, would like to actually have access to top-notch core facilities where we can, in fact, outsource we can send samples and they run them on a technology, proteomic sequencing, imaging, and we get data back. That's for an AI researcher, computational biologist, a dream. Combined with a wet lab that we have built that is a shared wet lab for all the dry labs. So we are kind of doing the reciprocal of what is often done. Lots of research is done at the wet lab, and then there's a computational core that can help you with the data analysis. We're going to turn this around. In this center, do all dry lab, all computational work with a shared wet lab, so that we can actually go through that shared wet lab to the core facilities, generate data, train our models, ask biological questions, or try to find them in the data, and then also go back to the lab and to a model organism to do the experiments. So that would be very interesting, particularly if it would cover several layers from the genome to proteins. You mentioned AlphaFold in the beginning, protein structure, protein interactions, and our first group leader is going to work on that topic, but also cells and organs with imaging techniques. And if we then have multiple groups covering these different layers, but with the same attitude, so very strong on computer science and on the AI methods, that would be the glue between people, but also I think between the biological layers. So explaining, it's still a massive, the core question of biology is genotype to phenotype. Here's a genome, what does it produce? Right, here's my genome, so can I predict from the genome sequence that I will be me, and from your genome sequence that you will look like you? This is still impossible. Right, so genotype to phenotype, and then one particular change somewhere, it increases the risk on this disease, or it makes you, this particular trait or this risk on disease. So that road between genotype to phenotype covers multiple layers, of course, not only gene regulation. And if we could cover more and more of that in our center with like-minded people who use computational tools first and foremost, but who also understand the data very well, who can run the experiment or make it happen, who can do the tests, and who are also having this open, vision on the process of doing science. That would be fantastic to welcome them. And I hope there's many candidates like that we could.
Yeah, it sounds like you've actually put a lot of thought into how you're going to design like the workflow and the shared spaces in the lab to actually increase the sort of collaboration or the ability of new group leaders to tackle the problems that they're interested in solving.
I did, and it's also based on some of the things that worked very well for me. The way that some ways I tried may not have worked well, but some ways I tried to combine experiment with compute worked well. And this is what I want to make available to other group leaders from around the world. And I think it is attractive because some postdocs who want to start their own group are now in a computer science environment and who have no access to running an experiment. So in the VIB, with all the expertise that is present in the whole VIB, and with the bridge that we want to offer them in our center, there's suddenly a play garden of model systems, diseases, from flies to zebrafish to chicken to human, of course, and it's all here, and technologies are here. So I think we have a good selling point, and so hopefully we can find some inspiring people from around the world to join us and attack the biology with AI.
Excellent. As we wind up here, something that was in my notes about you that I'd like to ask about is that you made a mod for the popular game Minecraft that taught users about DNA, genetics, Mendel's laws.
And still does.
And still does. Are you a gamer yourself?
Actually, not so much. I'm attracted to it. I like it a little bit, but I cannot call myself a gamer.
Did your kids have any input maybe?
My kids had input. My kids were playing Minecraft. My kids are more gamers. Some of them are more gamers than myself. But it was just a very nice idea at some point to develop a mod for Minecraft that would teach children about genetics, and we're still using that. And in the beginning, I was doing all these workshops with the kids in the Kinderuniversiteit, Dag van de Wetenschap. But then gradually, of course, I have asked people in the lab to join, and now people in the lab have taken over and are organizing the workshops. They go there, and new students, you often see that with master students, they are very interested to help with these workshops, to make the exercises. I've always had job students continue on developing the software, making it up to date, maintaining it, as well as developing tutorials and YouTube videos about it and spreading this and making it really useful for a workshop. And so we have seen hundreds already of kids passing through our workshop and coming out with sometimes just chaos and destroying everything in the Minecraft world that we built, or actually having learned something about genetics by crossing white and black chicken, or by extracting DNA from a chicken. And we have built a lab in the Minecraft and a sequencer, so you can extract blood from a chicken, sequence it, and then see the DNA of different genes. Yeah, and then if you follow the exercises and you are smart, then you can figure out by combining some recessive alleles in an egg. So you can actually take an egg, take the DNA from a chicken, clone it. So we teach children, in fact, of 10 years old about cloning. Change the DNA of an egg, and then you can make a chicken out of it. And if you have the right combination of genetics, there comes out a dinosaur. So this is at the end of the workshop that some kids are able to make a dinosaur out of chicken DNA. It's really nice, and there's a lot of good feedback on this workshop. It's still going. It still goes on. And it's always fully booked after like an hour on the website, fully booked. Yeah. Like Werchter tickets.
Yeah.
Really goes out fast.
Excellent. So before we finish the podcast, there's a question that we ask all the guests. It's the same question. Are you ready?
Kind of.
If you were to encounter an omniscient being and could ask that being one question, what would you ask?
What would I ask?
Mm-hmm.
I would ask, in which galaxy, or to how elaborate can my question be? Because
presumably the being is omniscient, so
I can
it can
ask
be complex.
Some stuff. It can be complex. So I would ask to explain or give me all the nitty-gritty details about life forms in other galaxies, likely, or on other solar systems in our galaxy. How far things have gotten, in history, like maybe millions or billions of years ago, that there's no trace anymore. And particularly if some advanced life forms exist, how this life is encoded, so that we could compare the way that it works here using ACGT sequences. All life on Earth, on our planet, all life uses DNA. And so we try to understand how it works. So obviously my question is, how in what other ways can you encode it, and how is it done in other places, and can we get some samples from there
to
do some single cell sequencing?
Give me some samples too, right? Yeah, excellent. Stein, thank you so much for the conversation. I really enjoyed it.
It was a great pleasure, Brad. Thank you so much.
If you want to hear more inspiring stories from our scientists at VIB, subscribe to this podcast. And if you like this episode, leave us a like or positive review. It really helps. Thanks for listening to Called to Science.